Previously, we talked about the exogenous lipoprotein pathway that involves chylomicrons. Now we're going to switch gears and talk about the endogenous lipoprotein pathway. This lipoprotein pathway originates inside the liver, and it's responsible for delivering fatty acids to extrahepatic tissue, for example, cardiac myocytes, skeletal myocytes, and adipose cells. So let's begin inside the liver. The liver is made up of liver cells we call hepatocytes. And inside the hepatocytes, we combine triglycerides with a little bit of cholesterol and phospholipids with a protein known as apolipoprotein B100, or simply ApoB100. And the purpose of ApoB100 is twofold. Number one, it acts as a core, as a scaffold to help produce the VLDL molecule. And number two, as we'll see at the end, it is utilized as a ligand to actually bind onto LDL receptors on hepatocytes. And we'll come back to this at the end. So inside the liver cells and inside the hepatocytes, we combine triglycerides, cholesterol, phospholipids, and a single ApoB100 protein to form nascent or immature very low density lipoprotein particles, VLDL particles. These are then released into the bloodstream. Now in the bloodstream, HDL molecules associate with these nascent VLDL molecules, and these HDL molecules give the immature VLDL molecules two additional apolipoproteins, so ApoC2 and ApoE. And once they receive the ApoE and ApoC2, they become mature and fully functional. Now these mature VLDL molecules are ready to give fatty acids to their target extrahepatic tissue. So the mature VLDL molecules circulate in the bloodstream until they get to the capillaries of extrahepatic tissues such as skeletal muscle, cardiac tissue, adipose cells, and so forth. Now, these cells produce and release lipoprotein lipase, LPL, an enzyme that associates with the endothelium of the capillaries. And so as these VLDL molecules travel across the LPL molecule, this apolipoprotein C2 acts as a cofactor to induce this enzyme LPL. And what LPL does is it breaks down the triglycerides into fatty acids and glycerol. Now, glycerol is a water-soluble molecule, and it simply remains dissolved in the bloodstream until it's picked up by the liver. And in the liver, the glycerol is used to help form glucose and other molecules. What about the fatty acids released by the breakdown of triglycerides? These fatty acids are quickly taken up by the skeletal muscle tissue, the cardiac tissue, and the adipose tissue. Now, in adipose tissue, we store it for later use. And in skeletal muscle tissue and cardiac tissue, the fatty acids are quickly utilized to help form ATP, and the ATP can then be used to help generate energy and basically stimulate different types of processes within the cells. Now, as we break down more of the triglycerides, this molecule becomes smaller and more dense, so it contains a relatively higher proportion of cholesterol compared to triglycerides. So remember, initially, the mature VLDL molecule is made up predominantly of triglycerides. But eventually, as many of the triglycerides are broken down, we're left with simply cholesterol. And so now we have a lot of cholesterol and very little triglycerides. And eventually, this apolipoprotein C2 is given back to an HDL molecule circulating in the bloodstream, and this gives rise to a molecule known as intermediate density lipoprotein, IDL. So IDL contains the apolipoprotein B100 that initially came from that liver cell, and also apolipoprotein E that came from circulating HDL molecules at the beginning of this process. Now the IDL can do one of two things. The first thing it can do is it can use the apolipoprotein E as a ligand to bind onto specific receptors on hepatocytes and other extrahepatic cells and be taken up by those cells. Or it can further give away an apolipoprotein, specifically apolipoprotein E, to an HDL molecule. And this forms low density lipoprotein, or LDL. LDL molecules contain a lot of cholesterol, and they contain the apolipoprotein B100. And these LDL molecules, because they contain so much cholesterol, they have very strong pro-atherogenic properties. And what that means is, if we have a lot of the LDL molecules circulating in the bloodstream, they can basically move into the wall of the vessel and become oxidized. And over time, this can cause atherosclerosis, so the creation and the buildup of plaque within walls. And that can cause things like coronary artery disease and peripheral arterial disease and so forth. Now, once we form the LDL molecule uses the apolipoprotein B100 as a ligand to bind onto LDL receptors present on hepatocytes. And once bound, we have a receptor-mediated endocytosis pathway that we'll talk about in the next lecture that is able to take up that particle into the cell, and the cell can break down the particle and reuse many of its units. Now, I want to come back to the liver for just a moment and talk about something called hepatic steatosis. Hepatic steatosis is also known as fatty liver, and this can develop in patients with obesity, in patients with diabetes mellitus type 2, and in patients who ingest chronic alcohol or even ingest alcohol acutely. So basically, in these conditions, we develop an imbalance between the synthesis of triglycerides in the liver and the synthesis and release of the LDL molecules. So in conditions such as obesity, diabetes mellitus type 2, and alcohol ingestion, we essentially have more production of triglycerides than production and release of the LDL molecules. And so what happens is, if we have more production of triglycerides than normal, then we essentially produce and store too much triglycerides in the liver, and that can cause the liver to actually become fatty. And that's why we call this fatty liver or hepatic steatosis.